everyone, welcome back to We Smart Street Conference video series. We hope you enjoyed the previous video. And in this video, we will show you the talk from Dr. Rachel Cavill. So she's an assistant professor in the Department of Data Science and Knowledge Engineering at Maastricht University. And in her talk, she talked about how researchers can responsibly present the research and outcome to the scientific community and the general public. So she took some examples from biomedical domain, but the principles she raised can be applied to other domains as well. So if you are interested, continue watching that.
So next up, we have a Tech Vision talk by Dr. Rachel Cavill. Dr. Rachel Cavill is an assistant professor in the Department of Data Science and Knowledge Engineering, part of Maastricht University. And after receiving her master in mathematics and computer science from the University of York in 2002, she continued at the same institution and received her PhD in 2007 by developing bio-inspired machine learning algorithm. She transitioned into the field of bioinformatics during her postdoc years. And during this time, she spent 4 years working at Imperial College in London, focusing on the analysis of omics data, in particular the integration of data sets acquired using different technologies on matched samples. This theme of data integration takes a broader form in her current research, in which she and her team explore how machine learning and data science approaches can allow us to integrate biological data sets from different sources. So today she'll give a talk about reporting robust research results, perspectives from data science in the biomedical field. With that, I would like to give the floor to Rachel. Yes, there she is. Welcome, Rachel.
Thank you. Can you hear me?
Yes, I can hear you.
Good. I'm going to be talking about reporting robust research results. And I want to start with how we all start when we do data science, is that we have a biological data set. So when we get a data set, whether it's biological or not, we always need to start from the questions that we've got. And one of the problems that I have as a data scientist is that most of the time, I don't always want to start from the questions that I've got. I want to start from that cool tool that I know how to use, that fun thing that I've just learned about. And this leads to the problem in this picture that I'm hitting a nail with a hammer, but it's not actually a nail that I'm trying to hit. I'm actually trying to hit something else, or it's all going in one key and everything goes wrong. So I really want to encourage you that whenever you get a data set, you start from the questions that you've got, not from the tools that you have or the techniques that you know, the algorithms that you're familiar with. So what kind of questions should we be starting from? Well, one of the common questions is which samples are most similar to each other. And of course, when we've got that question, we should be going for an unsupervised technique, probably like clustering. So we might have hierarchical clustering or k-means, or even something like principal component analysis that looks at the variation in data set and projects it into a lower number of dimensions, some kind of dimensional reduction technique. A second question we could answer is, can I predict new samples? Pretty standard machine learning question. And for this, we would end up with supervised machine learning. So random forest, neural networks, support vector machines, all of these type of methods, and a whole lot more. But then we get to the question of what variables are important. And this is one I want to linger a bit more on, because this is a question that I see a lot of people using machine learning for, especially in the biomedical domain. People say, well, I'm going to use some machine learning and I have no interest in predicting new samples. I just want to know what variables are important in my data set. And I'd like to suggest that a lot of the time, machine learning isn't the right tool to answer that question. And that sometimes traditional statistics might be a better tool to answer that question of what variables are important. If all you're interested in is what variables are important and not predicting new samples, And especially if you're interested in what variables are important on their own, then traditional statistics can give you those answers and can give you it in a more robust, a more reproducible, and a better way than machine learning a lot of the time. So whenever you get this what variables are important question, I'd really like to encourage you to think about, why am I going for machine learning here? To answer the question, what is machine learning going to give me that statistics can't or won't give me? What's it going to add? Why is machine learning going to be better than statistics for answering this question with my particular data set? And what might machine learning miss? So let's think about, if I wanted to build a single decision tree on some data, and I've got two variables that are sharing the same information. They have almost the same values for everything. Well, my decision tree is only going to pick one of them, and it's going to pick the one that's best on my training set. It's not going to be worried about the fact that the other one is also good. So if I use machine learning, and in particular that single decision tree, to answer the question, what variables are important in my data set, I'll only get one of the two, but actually they were both pretty important. They're both pretty distinguishable between the two groups, and that's why they would be important in the tree. It's just that we only need one of them to be able to answer the question. And this happens a lot with biological data sets. We get a lot of variables that are collinear, they're correlated to each other. They contain the same information. And with some machine learning methods, this can cause us issues, that we highlight one of these and we ignore the others because we only need the information from one. And it leads to potentially a not so robust classifier as well. So that's another issue. But it leads us to a bias that we see certain variables with machine learning that we wouldn't, but if we did statistics, we would get a better set of variables. There's been an example in breast cancer, for instance, where people were building machine learning programs, algorithms on predicting breast cancer stage or predicting the presence of breast cancer. And this was quite a few years ago in the literature. Lots of people were doing it on different data sets from different hospitals, and they were all finding that different genes were important, different variables were important. And there was a lot of questions being asked. Well, why aren't we seeing the same variables coming up again and again with machine learning from different data sets? What's going on? A counterexample. I've got a data set at the moment that I'm working with a bachelor's student and the Department of Biochemistry. So the Department of Biochemistry have given us this data set, and it's got just 5 variables, 5. So it's not big data here in that direction, but it's a hypercube of data. What do I mean by that? Well, we've got 5 variables, and each of those 5 variables were measured at 5 time points. And each of those 5 variables and 5 time points were measured with 4 different interventions. And each of those 5 variables, 5 time points, and 4 interventions were measured on 3 different surfaces. And all of that was done for 13 different healthy donors of blood. So they give their blood, and we do all of these experiments with their blood. And this is a data set where there's some interesting things going on, because we want to know which variables are important, but we also know that there's going to be non-linear relationships between the variables. And we want to know not only which variables are important, but at what time points, for which intervention on what surface. And of course we can do, and we have done, the basic statistics here. So our questions are, what changes after an intervention? Well, that basic statistics is probably the best thing to look at, because we could just compare the parameter before and after intervention, or with and without intervention at that time point, and see what's going on. The other question we've got is, which combination of parameters on what surfaces and at what time points can we measure to distinguish between the interventions? And that sort of combination idea is where machine learning can give us an added bonus, because it can take combinations into account in a way that traditional univariate statistics doesn't. the other advantage that machine learning can give us is it can give us nonlinear interactions as well. The tricky thing here with selecting variables is that we don't just want to select the variables that are the absolute best, because if I would need to measure in the lab, in the clinic, I need to measure parameter 1 at time point 1 on surface 1 to get some information about this intervention. But the other variable I need to measure is time point 5 on surface 3 at parameter 2. Then that's going to be a lot of experiment. But if I only need to measure a couple of parameters at the same time point on the same surface, that's going to be a lot less experimental stuff to measure. So we're trying to work out at the moment how we can sort of incorporate that into our machine learning. How can we give our machine learning a bias towards wanting to give us parameters that are measured at the same time point on the same surface, or perhaps even a different time point but on the same surface, so that we can minimize the lab work that would need to be done to predict future patients. So we've decided to use machine learning after all. To make our science reproducible and robust, we of course need to make sure that our models don't overfit our data. And hopefully this is a concept that you're all familiar with. And of course, as Anna just said in the presentation about the datathon, you need to split our data into test, training, validation sets. Just want to stress that. There's also an interesting field in machine learning called conformal prediction, where you look at trying to learn the actual p-value of precise prediction of how accurate your predictions are, and the precise p-value of that. And for that, one of the methods you can use is to keep another set back for conformal prediction. Of course, you then need even more data. So you need your test set, your training set, a validation set, and then also a set to learn the conformal prediction. One of the really key things here with your test and your training set is information leakage. And information leakage is when the training set gets information from the test set. So our test set or our validation set isn't independent anymore. And the problem here, especially with biological data sets, is that most of the common pre-processing data sets that we do can induce information leakage. So some normalizations, if we normalize all our data together before splitting it into test and training, so many normalizations will give us some information leakage. That our test set will have some information from the training set, and our training set will have some information from the test set, because we've normalized them together. Filtering of unexpressed values, even that could give us information, because if by chance we filtered a gene, we filtered genes that are unexpressed in 50% of the samples, but if by chance that gene was expressed in all the samples that end up in our test set, and not so many of the samples that end up in our training set, well, it wouldn't be in the training set if we just did the filtering on the training set alone. So that can bring information across into our training set about the test set. Imputation of missing values. If I use the test set when I'm imputing the missing values in the training set, I'm going to have a lot of information leakage. And all of this means that our results are not going to be robust, they're not going to be reproducible, and they're not going to be as useful to the community as we hope they would be. So how are we going to prevent information leakage? Well, the best way of preventing information leakage is to treat our test set from the raw data point onwards as if it's new data we want to predict. So we don't touch it. We just pre-process our training set together and then we go on. The problem, of course, is that a lot of the time the raw data files are enormous, so we tend not to share them. We tend to share the pre-processed data instead, because that's, you know, we want to be fair, we want to be open about our science, we want to share our data, but we also have file size limitations. And so a lot of the time, the only data you can download if you're using someone else's data set is the pre-processed data. So how can you control for information leakage then? And I don't have those answers, but I think that's an interesting question that we need to address within the field. And when we evaluate performance, of course, our training set performance doesn't tell us about future performance. Computers memorize data if the algorithm allows it. So being able to recognize what they've already seen, don't care about that, you know. But that doesn't mean that training performance should be ignored. So a few years back, I remember a lot of people were reporting their training performance and perhaps even averaging the training and test performance of the headline performance. And I was going, no, don't do that. Now I see almost the opposite, that people don't even talk about the training set performance. They don't even tell us, and they don't even look at it sometimes. Yet the training set performance tells us so much. It tells us about when it's better than test performance. Might have overfitting if it's a lot better. It tells us about which classes are well and badly predicted. I mean, sometimes you get an imbalanced data set. A lot of the time in biological data sets, they're imbalanced. And you see that the training set being so good, you know, you might have a predictor that can predict 80% of the time, but it's only predicting one class. So an example of that, I can build a classifier, I say build in inverted commas, that's going to predict the COVID-19 status of people taking COVID-19 tests recently when I made these slides with 90.3% accuracy. And my classifier is really, really simple. My classifier is everybody's negative. Everybody doesn't have COVID, because it turns out that only 9.7% of people who were tested for COVID between the 15th of February and the 21st of February were positive in the Netherlands. So I can get 90% accuracy just by saying that everything is negative. And I've seen it quite recently, quite a few examples of people giving me classifiers that they say, this is 80%, this is 90% accurate. And then I look at the training set performance and it's not getting any of that one class right. It's just predicting the other class. In this case, you really want to look at doing some balanced accuracy or under or over sampling, because otherwise you're going to go completely wrong. And I want to think a little bit about variable selection for a couple of minutes. We're about to be stranded on a desert island. I've got a big pile of books there, and I'm going to sort them with my favorites at the top of the pile, but they've got 10 copies of my favorite book. And so I'm only going to carry 10 books. What am I going to do? Do I take the 10 copies of my favorite book, or do I sort through and take some other books to give me more interest? Well, I'm going to sort through and take other books to give me more interest. And that's the problem with these sort of fixed criteria selection, the problem with selecting variables. There's different ways of doing it. We can do a fixed criteria selection. We do this a lot. We just say a p-value, correlation, fold change cutoffs, and we get a nice list, which is really useful for biological interpretation. But it might not be so useful for building our predictive models, because all of these variables are going to, or many of them will carry the same information. They'll be correlated with each other, and variables that have other useful information but don't meet these cutoffs have been thrown away. We can do embedded selection, which is how some machine learning methods, random forest decision trees, do it. Inherently select variables. Forward selection, select the best, keep going, or backward selection, start with everything, remove those that aren't useful. Problem with backward selection is it's really slow. And the other problem is we've measured thousands of variables. There's so many different sets that have the same performance. So a bit of an example with some variable selection. This was a study I did many years ago now, about 10 years ago. We looked at transcripts and metabolites and growth inhibition in cancer cell lines, and we're looking for genes and metabolites that are associated with platinum sensitivity. So we take the growth inhibition across, we've got a value for 60 different cell lines, and we've got a value for each gene for 60 different cell lines. So we look at the correlation between those two values, and for each platinum drug, we get a list of genes, and we do the same with metabolites. So the first thing we did was that we put in random lists, the same size as the real lists, and we saw whether we got similar results. And yes, we see that we get a lot more results with the real results than we do with the random ones. So our false discovery rate for that model is 0.2%. That's brilliant. Means we're really not seeing much false discoveries because we don't see much when we put in random lists. The next thing we did was we put in random lists at the pathway level. So we put in random lists of the pathway, and we looked at how much overlap do we see between the 4 drugs. Again, really small amount of overlap. That was great. The third one we did was that we permuted the labels from real pathways for each drug before doing the integration between the omics. And then we see it wasn't quite so significant. 2 of the drugs had more pathways with real data, whereas some of the other drugs didn't. But overall, this was a really good way. This putting in random lists was really useful.
Rachel, we need to wrap up.
So remember that when you do unsupervised machine learning with selected variables, it becomes supervised. So here I've got some random data on the left, and then I did some fixed criteria variable selection, just picking the variables with absolute correlation bigger than 0.5, and I get this horrible plot with just the 2 things, what really separates brilliantly. Remember, because this is random data, there's no other patterns in the data other than the thing that I've just selected for. So it looks even worse than our real data would look. So to summarize, don't just use machine learning because you can. Think about why you want to use it. Watch out for information leakage. And if you're interpreting the variables selected by a model, think about that tells you about what the model uses to predict. It may not tell you everything about what variables are different between the groups. And that was the point I was trying to make at the start with the variable selection. Thanks for the opportunity to talk, and hope to chat to you all some more later in the panel discussions.
Yeah, Rachel, thank you very much indeed.
Thank you for watching. We hope you enjoyed the video. So if you want to watch other videos, please check our playlist.